but Pittsburgh's kind of known, in large part due to UPMC, nationally as a city that does medical research. So, say again? Well, yes, Jonas Sarks. Yeah, that was way before, but he had a Pitt connection. Yeah, the polio vaccine was developed here. And there's all kinds of fascinating companies, people making real money doing this kind of work in Pittsburgh. All right, let's talk about genetic engineering. Remember, everything's here that needs to be here, but we're going to mark it up, right? And then we're going to submit this to campus. So genetic engineering, first of all, you need to know engineers don't just drive trains, right? Engineers are all about solving problems. My wife's actually an engineer, and I create all kinds of problems for her to solve in our daily lives. But that was a joke. Solving problems are what engineers do. So there's all kinds of engineers. There's mechanical engineers that solve motion problems. There are civil engineers that design bridges and road systems and intersections. There are chemical engineers that make large-scale chemical reactions happen. And there are genetic engineers. And so genetic engineers basically are using DNA to solve those problems. And so that's really what genetic engineering is all about. It's using DNA to solve problems. And there are no shortage of problems. And so we're going to look at 3 problems. We've already talked about a couple. Organs, right? The organ shortage is a huge problem. We're going to look at 3 examples of problems that are very different from each other, where the solution for all 3 of these is manipulate the DNA. The first one is crime scene investigation, which you guys probably know as CSI, right? That's how they always refer to it on the crime shows. CSI. And the technique that engineers have developed for identifying biological evidence at a crime scene is called DNA fingerprinting. So we can find a blood sample or a tissue sample or saliva or even skin at a crime scene, and we can use this technique to identify or match it to a victim or a suspect, right? And this is a powerful technique. The second problem is in the field of agriculture, growing stuff. And so we have genetically modified many organisms to make them better food. Now, we have to maybe put better in air quotes, right? Because there are people that don't think that that's better, but there are some, farmers in particular, that do enjoy higher yields and higher profitability due to genetically modified food. A lot of the food that's available to us in the grocery stores nowadays is genetically altered. It's genetically modified, and it's not just by selective breeding nowadays. It's by actually manipulating the DNA. And then the third field that we're going to think about is medicine. And there's a technique out there that's becoming more and more common called gene therapy. And there are individuals with a certain type of blindness that have been able to have their vision restored due to gene therapy. Really powerful technique. And basically the idea behind gene therapy is, well, if a person has a condition because their DNA is messed up in some way, why don't we just give them the correct DNA sequence? That's basically what gene therapy does. Go ahead, Eliza. Yeah, I'm sorry. Why did I? Oh, because Eliza in my homeroom was having trouble scheduling. Sorry, Isabella. Oh, really? Wow. It was fine before? Oh, wow. Well, that's exciting. Fun. All right. Cass. Yes, go ahead. There, you got it. Hey, Canyon. Yep, you're right. Yeah, so that's they call those designer babies. And that's something that is possible nowadays. Of course, there's all kinds of moral and ethical questions to be thought about with that. That's not something we've done in our country, but the first designer babies to be published did happen 2 years ago in China. So China was the first person to genetically modify a baby, and they basically gave them a gene that would prevent them from being able to ever contract the AIDS virus. So it was viewed as a good, positive change for humanity. But still, there was a lot of upset people. They were like, you shouldn't do that. You don't have authority to do that. Does anybody have the authority to say that you genetically are inferior? Right? I mean, it's a slippery slope between saying that and, all kinds of other, racism and all kinds of things, right? All kinds of prejudice and things like that. So there's a lot of questions. Of course, that wouldn't be the goal of designer babies. The goal in designer babies is to genetically modify babies, Jenna, so that we can prevent a disease from happening. And so we have the ability to do that right now, but there's all kinds of ethical questions. Do we have the authority to do that in a country like ours where we value human rights and individuality so much? But yeah, it's a good question. Jenna, what were you going to say? Oh, no. Jenna.
Yes, what about dog breed?
Yeah, dogs we can do. I mean, this sounds a little bit mean, but we can do many things to dogs that we would never do to humans, right? And
yeah.
Yeah.
Like Shishan.
Yes, definitely. Okay.
Well, and that's what Cookie is.
All right. What we're going to do today, I think, is tackle the first problem. So let's look at this first problem. This is called DNA fingerprinting, okay? DNA fingerprinting. The first thing we need to say is that this is not fingerprints. They're making an analogy when they say this. So it's not actually fingerprinting. And I'm drawing a blank here, guys. Are we doing a DNA fingerprinting activity today? Was that on the agenda? It is. Okay. We got a motor. So everyone has a different order of nucleotides in their DNA. We said most of our DNA is the same. That's what makes us a human and not a dog or fish, right? But there are obviously differences, right? The DNA is what's causing us to have different heights and different hair and different eye color and all those traits that make us unique. Okay? So we're not actually fingerprinting, but just like our fingerprints are unique to us, right? Our DNA is unique to us. And so we can use DNA that's found in biological material.
Mr. Kramer, I'll be right back. I got to put one of my dog's bowl.
We're going to keep teaching a little bit because we got to get through this content. So just hold on to that question, Jen, and we'll get you, okay? Any biological material that we can use to identify a person or to identify a suspect or a victim. Not to be morbid here, but sometimes when we find a victim, it's been a while, if they've been murdered or something like that, and the body starts to decompose, we can use this technique to identify who that person is very precisely. And so this is basically the goal of DNA fingerprinting, is to identify people. If we find some hair or saliva or even skin at a crime scene and we think that was there because of the suspect, well, then we can use the biological information from that tissue and try to match it to a suspect. And so we can use a lot of different things as evidence of our DNA. We're constantly giving off cells, right? You look at the school at the end of the day, there's huge dust bunnies every day that are in our school, and that's all skin and hair and things that have fallen off of us as we go through our day. So how does this work? It's got to be, in order for it to work for our government, it has to be cheap and effective. And that's what it is. The first thing we do is we use enzymes that are called restriction enzymes. You guys remember, enzymes are tools. I like to think of restriction enzymes as kind of like scissors. They're scissor tools. Okay, so every tool has a shape. Think about the shape of scissors. And of course, scissors cut stuff, right? And so restriction enzymes will cut the DNA at specific nucleotide sequences. It doesn't just cut the DNA randomly, it cuts in specific places. Okay? So for instance, let's just do a GGCC. Every time the DNA has a GGCC, our restriction enzyme will make a cut there, right? And then there would be other restriction enzymes that could do cuts at other specific patterns of nucleotides. And so when this happens, you get DNA of different sizes because everybody's code is different. You get different pieces of different sizes. And again, we don't have to know all the nucleotides in a person to do this. That's the beauty of this, is we just add the enzymes, they cut wherever they find a specific nucleotide sequence, and it creates DNA of different lengths. The next thing that we're going to do is we're going to separate those DNA of different lengths. By the way, you'll notice I bolded a couple of words. Those are the summary words for each step. So step one is all about cutting. So we first cut the DNA. The second step is all about separating those pieces of DNA. And so the way that we do that is using what's called gel electrophoresis. This is like a little molecular obstacle course. You guys have watched American Ninja Warrior. You know what we're talking about, right? In American Ninja Warrior, there's an obstacle course. And does everybody go through American Ninja Warrior at the same pace? No, right? And sometimes it's the big, heavy guys that don't make it, right? They're ripped, but they're too heavy to make it over all the obstacles. The same thing is happening in this gel electrophoresis. The DNA, depending on its size, will move through this jello-like material faster or slower. Small pieces go fast, big pieces go small, or go slow, I should say. So maybe we'll do small and fast. Let's connect those two ideas. Small is fast. Big, I guess I should add the word slow. Big ones go slow. I like to think about drag racing, right? If there's a Porsche and a tractor trailer, right? The Porsche is going to go very quickly, right, because it's small and powerful. The tractor trailer is powerful too, but it's going to go much more slowly through the gel. And so we're separating the pieces of DNA based on their size. Third step, the word that's bold is compare. We're going to compare. When we separate the DNA, it will collect in certain places depending on the size of the pieces, and this creates a banding pattern. I think when I think of banding pattern, think of barcode. You guys are all familiar. Everything that you buy at the grocery store has a barcode. That's what they use with the laser at the checkout line. Yeah, even your Chromebook has a barcode that makes it a unique individual, or a unique Chromebook, so they can keep track of them. The barcode is a pattern of lines that's unique. So the barcode that's on Cam's Chromebook is a different barcode than the one that's on Lindsay's Chromebook, right? It's a different pattern. So the spacing of those lines is unique. And so that's what a DNA fingerprint actually is. It basically creates a unique barcode that is unique to you. And a different person is going to have a different barcode. And so we can compare these barcodes and say, that's a match, right? So now this person, we've determined that your skin was found at the crime scene. We know you were at the crime scene. Stop denying it. What were you doing there, right?
And Mr. Kramer,
What, Jenna?
there's a number.
A number, 5 or 6.
No, there's an actual number that is below the Chromebook.
Oh, yeah, that's true.
Below the Chromebook's code.
Too. You're right. Anybody need more time? Brandon, you good? No, I'm not. You're not good. Okay, I'll wait. That's okay. I'm sorry. You've got a technique, though. I'm glad you got that. I didn't wear a tie today, though. Amanda, you don't wear it. It's fine. You can't tell because the mask is on. No, I just look more. All right. Everybody always wants to take pictures of my outfits. They're so creative. All right. Any more time? We good? All right. Here we go. So here's a nice little visual representation of what we've been talking about. And by the way, if you're interested, there's a class here at the high school called Bioinformatics where you can do this. They do this in Bioinformatics with Dr. Stein. But let's take a look here. We'll just label a couple things. This is the gel. Remember, it's the obstacle course, okay? And this, of course, represents our DNA cut to different lengths. Okay, so we restrict the DNA, we cut it up into different sizes, and we insert it into these guys. These are what we call the wells. Can you imagine this to kind of look like a well in profile? It's a well, that's where you put the DNA in. And so the DNA travels through the gel from the well side to the other side. And the smaller and shorter pieces go fast. So let's label these ones small pieces. They're like the Porsches, right? And then let's label these guys here. These are the long pieces. They're like the tractor trailer that's still, getting into second gear. And so the whole thing together, right? Maybe I'll circle this one. That's a DNA fingerprint. It's a banding pattern. It's not an actual fingerprint, right? But it is unique to a person, just like your fingerprints are unique to you. Yeah. DNA fingerprint, and then on the left, long pieces. So the pieces that are closest to the well, they're going to be the ones that are the longest. They're like the tractor trailer. They're not going to be able to go through the gel very far. The small pieces quickly go through the gel, and so they separate out according to their size. The pattern is what we call the DNA fingerprint. And so each and every one of us has a unique barcode, has a unique DNA fingerprint that makes us different from everyone else's. This technique is really cheap and pretty accurate. Usually when lawyers argue against this kind of data, they usually talk about how the evidence was mishandled, right? They try to draw doubt or skepticism that the lab somehow messed up the technique, because they know that the actual evidence is pretty significant. All right. We're out of here at, what did we say, 9:10, okay. Okay, guys. Let's see. What should we do? Should we continue through the notes, or should we pause and do this activity? I don't really know, Miss Priscilla.
Let's pause.
Yeah, I would. Yep, that's good. All right, so let's set this aside. Let's